Christo is currently a researcher at the Coastal Systems Group in CSIR, and he's based in Stellenbosch in Cape Town. As Dion alluded, he basically developed regional and optimization, sea surface temperature, and chlorophyll A concentration Earth observation products that allow for accurate, high resolution, temporal, and spatial monitoring and analysis of biophysical variability along the Southern African shelf. And for this area, that's of great importance because this is where everyone gets those beautiful pictures where they show you upwelling or they show you chlorophyll A concentrations to show you that this is a highly productive system.
I would like to introduce you to some South African perspectives on utilizing Earth observation data in practical regional applications. My lecture is entitled Addressing Earth Observation Challenges in the Benguela Upwelling System. I will demonstrate how Earth observation products developed at the CSIR is used to detect and monitor harmful algal blooms in support of South Africa's small-scale fisheries and abalone aquaculture industry. This presentation reflects work done by the CSIR Marine Earth Observation team, together with my colleagues Dr. Marie Smith and Dr. Stuart Bernard. Allow me to expand on the South African government's undertaking to address poverty alleviation, economic transformation, and a measure of self-determination for our vulnerable coastal communities. South Africa has an underexploited marine exclusive economic zone that exceeds the size of a terrestrial territory and manages a coastline larger than 3,000 kilometers. In July 2014, the South African government launched Operation Phakisa, an initiative aimed at fast-tracking the delivery of some of the priorities outlined in the National Development Plan. The National Development Plan is the country's socio-economic development blueprint, which enjoins citizens to create a better life for all citizens in an inclusive society. Phakisa means hurry up in Sesotho, one of South Africa's indigenous languages. The initiative is modeled on Malaysia's Big Fast Results methodology and was designed to fast-track the implementation of solutions critical to socio-economic development issues. To facilitate the management and growth of a blue economy, the South African government initiated the development of the National Oceans and Coastal Information Management System, referred to as OCIMS. In June 2015, the Council for Scientific and Industrial Research was nominated as a service provider to facilitate the implementation of the project and to co-develop OCIMS. Considering the vast expanse of our marine exclusive economic zone, Earth observation data is recognized as a key resource in the development of decision support tools for management considerations. OCIMS seek to integrate information from satellite, in situ, model, and lab-based sources into user co-developed decision support tools that address early warning support, operation support, compliance and enforcement, as well as planning and assessment within South Africa's coastal and marine exclusive economic zone. The project aim is to produce information that will have a measurable impact on the South African socio-economic landscape by identifying, interpreting, and monitoring environmental factors that affect the sustainable exploitation of living and other marine resources. West Coast rock lobster, also known as crayfish, is an important resource to small-scale fishers on the west coast of South Africa. Here, coastal communities rely on seasonal catches to earn a livelihood. During February and March 2015, large marine mortalities resulted in the removal of 415 tons of rock lobster, 21 tons of mussels, and various fish species from the beaches within the St. Helena Bay area. The huge loss of valuable marine resources was due to the presence of large harmful algal blooms, also known as red tides. Although red tide organisms are known for the toxins that they produce, most of the ecological harm results from the high biomass that they are able to achieve. In the southern Benguela, huge blooms sometimes cause anoxic conditions followed by the subsequent production of hydrogen sulfide, leading to high mortality in several species. Aquaculture farming offers significant potential for rural development and economic advancement, especially in marginalized coastal communities. The advent of Operation Phakisa has provided significant stimulus for growing and transforming the aquaculture sector in South Africa. Legislative reform was accelerated, and the Aquaculture Development Bill was approved by Cabinet in 2018. An enabling regulatory environment was fostered through the creation of an interdepartmental authorizations committee to coordinate aquaculture applications and approvals. The consolidation of funding from several government departments into the Aquaculture Development Fund supported the growth of the existing aquaculture concerns and the initiation of 17 new aquaculture projects since 2014. Implementing Operation Pakise initiatives within the aquaculture sector has proved to be quite successful, providing employment to rural communities and increasing the overall South African aquaculture production, thereby generating considerably more revenue within the national economy. Currently, aquaculture farming includes finfish, mussels, oysters, as well as the land-based farming of abalone. Farms are interspersed within coastal communities along the South African coastline, from Honderklip Bay on the west coast to Richards Bay on the east coast. Globally, South Africa is the third-ranked exporter of farmed abalone. This sector of South African aquaculture farming presents the greatest potential for growth in the medium term because of the product's high market value, while also generating approximately one job for every ton of abalone produced. In 2016, South Africa produced 1,500 tons of abalone, which was only 1% of the global supply. But the sector has seen significant growth and estimates that it could realize export numbers of 6,800 tons per annum by 2025. Although there are 12 abalone farms along the coastline between Honderklip Bay and East London, the heart of South Africa's abalone farming industry is found in Walker Bay. Land-based abalone farms circulate 1.2 to 1.5 million liters of seawater per hour, and one of the biggest threats to production is posed by harmful algal blooms in the adjacent marine environment. In January 2017, Walker Bay was inundated by a large red tide that impacted three land-based abalone farms, causing the death of several million abalone and financial losses in excess of 50 million rands. All the aquaculture industries are to some extent susceptible to the negative impact of red tides, whether directly through the assimilation of toxins, mechanical damage, or indirectly through the anoxia from bloom collapse. The seasonal occurrence of red tides is a common theme within the southern Benguela upwelling system, and their effects are felt from St. Helena Bay on the west coast to Algoa Bay in the south. Red tides are most common during the latter part of the upwelling season, when the contribution of dinoflagellates to the phytoplankton community is greatest. Under conditions of upwelling, the dinoflagellate population tends to accumulate offshore, in a region of convergence created by a narrow band of upwelling and the core northward surface flow. Stratification is recognized as a precondition for the proliferation of red tide organisms, which are considered to be particularly susceptible to turbulence, but well adapted to lower energy stratified conditions. During periods of wind relaxation or reversal, shoreward accumulation of dinoflagellate blooms occur, often followed by the general southward progression of these blooms in the presence of an inshore countercurrent. Early detection of bloom proliferation and subsequent identification of the dominant species is essential for the protection of community health and economic activities in regions where red tides commonly occur. Sea surface temperature and chlorophyll A concentration are key Earth observation products that can be used as decision support tools for the early detection and monitoring of red tides to mitigate against the harmful impacts associated with their occurrence. This sequence of Sentinel-3 images depicts the shoreward accumulation and southward translation of a harmful algal bloom event in the St. Helena Bay region on the South African west coast during February this year. Characteristic warming of sea surface temperatures is seen along the coast as upwelling is suspended. This signifies stratified conditions and a high probability of potential bloom initiation. A large blob of high biomass is observed close inshore by 12 February and translates southward from Elands Bay to Dwarskerspos. The bloom is persistent in the southern St. Helena Bay region for the next 2 weeks, oscillating on or offshore depending on the prevailing wind conditions. Further northward, there is some indication of bloom activity near Doring Bay, with a significant threat posed to the abalone farming activities on 23 February. Farm managers at that facility needed to consider whether to close the intake of seawater and recirculate the water within the system until the bloom had dissipated. Standard cloud flagging options for MODIS SSD either removes too many valid ocean pixels, as seen in Figure B, or retains the pixels representing cloud edges, as seen in Figure C. The CSIR Earth Observation Group developed an SSD cloud flagging procedure that reduces the impact of cloud edge pixels on single overpasses, as seen in Figure D. Their presence is further reduced by generating daily averages of declouded single overpass scenes from both MODIS sensors using daytime and nighttime data, as seen in Figure E. The final product retains more SSD pixels and accurately represents the physical phenomena in the southern Benguela, while retaining the strong gradients associated with the edge of upwelling plumes and the good object current, as seen in Figure F. This Benguela optimized SSD was validated against the GIRST multi-scale ultra-high resolution SSD product and in situ data. SLSTR data will be added to the Benguela SST product to maintain the time series continuity. Under most conditions, any water-leaving radiance contribution in the near-infrared is considered as radiance contributed by aerosols and subsequently removed from the empirical blue-green modus chlorophyll product. This assumption holds except in coastal environments with large reflectance contributions from surface sediments or very high biomass biological blooms. Since the empirical chlorophyll A algorithm fails to produce an accurate result under high biomass conditions, it is necessary to switch to an alternate chlorophyll A retrieval algorithm when encountering these pixels. Satellite-derived fluorescence line height is mainly correlated with chlorophyll A concentration through first-order linear functions at low chlorophyll A values or power functions at higher chlorophyll A values. The CSIR Earth Observation Group derived chlorophyll A concentration from the MODIS normalized fluorescence line height product using a power function and used the result to develop a switching algorithm for MODIS to accurately track high biomass values associated with harmful algal blooms. The Benguela optimized MODIS chlorophyll A concentration product showed increased pixel retrieval and an improved correlation with higher biomass values when validated against in situ data. The empirical blue-green algorithms used to retrieve chlorophyll A concentration for the MERIS and ALCHI sensors present similar accuracy issues when high biomass pixels are encountered. The CSIR Earth Observation Group developed a novel technique to apply and blend two different chlorophyll A algorithms, an empirical blue-green algorithm for low to moderate biomass and a red near-infrared band ratio algorithm for moderate to high biomass waters. The blending method is based on the 708665 nanometer reflectance wavelength ratio, where the blue-green algorithm is applied when the ratio is less than 0.75, and the red near-infrared algorithm is applied at values greater than 1.15, whilst the two are blended between these values. The Benguela optimized MERIS ALCHI switching algorithm showed superior performance in the southern Benguela compared to any single algorithm when compared to in situ data. OSIMS is now in the fifth year of its implementation, and various decision support tools were developed in a co-design process with marine stakeholders for distribution via the OSIMS portal. While OSIMS supports the general socio-economic potential and ecological conservation of South Africa's oceans and coasts, the harmful algal bloom decision support tool specifically provides information to the aquaculture industry and environmental managers, allowing the timely implementation of appropriate mitigation steps. This is an example of the Fisheries and Aquaculture Decision Support Tool. Risk assessment and monitoring are based on quantified understanding of bloom dynamics, hypoxic impacts, and Earth observation capabilities. Maps of various ocean color-derived phytoplankton biomass proxies and sea surface temperature are used together with wind, current, and sea state data to monitor the initiation, proliferation, and movement of blooms. Several coastal locations are listed in the table to the right of the map, with high-risk harmful algal bloom development indicated by red dots in columns representing the last 7 days. The user can select their chosen product from the thumbnails below the table and navigate to their region of choice using the large map. There is considerable overlap between South Africa's OSIMS implementation to facilitate blue economy developmental goals and the regional strategic aims of member countries on the Southern African subcontinent. GMS in Africa aims to promote the development of local capacities with respect to institutional, human, and technical resources to facilitate access to and exploitation of Earth observation-based services. These services should be implemented on an operational basis for sustainable development in Africa. In 2017, the CSIR and regional partners successfully submitted a proposal to implement the Africa Marine and Coastal Operations for Southern Africa project in response to a call from the African Union Commission in the GMS Marine and Coastal Thematic Area. This project is referred to as Marco South. There are 9 partners in the Marco South Consortium, representing the interests of Angola, Namibia, South Africa, Mozambique, Tanzania, Kenya, and Mauritius. Since there is considerable synergy with OSIMS, the Marco South project will effectively provide a platform for the regional expansion of the services developed through OSIMS. Regional initiatives include supporting the development and operations of artisanal seaweed farming and other small-scale aquaculture in Tanzania and Kenya, supporting small-scale fishing communities through improving fishing effort and safety across the region, supporting national sea rescue institutes regionally, supporting the regional small and large pelagic fishing industries, and supporting the aquaculture industries across abalone, bivalve, shellfish, and finfish sectors through bloom risk, site selection, and operational optimization support. Marco South and OSIMs follow a similar co-design process, where there is active engagement with users during workshops, facilitated by quantitative mechanisms for user co-designed systems. The CSIR has established central data acquisition and processing facilities with the aim of serving user-optimized products as lightweight, high-value web and app-based services. Partners and users can determine their level of engagement to be as pure consumers, with a desire for the EO tools to serve specific applications, or as high-level researchers involved in marine ecosystem management, monitoring, and investigation. I would like to conclude by confirming that the OSIMS tool is already making an impact in several spheres of our blue economy management. In 2019, a large harmful algal bloom in Walker Bay resulted in no mortalities due to improved fire mitigation measures and information from the aquaculture and fisheries tool on bloom location and operational planning. Although the detection of high biomass waters in the southern Benguela is a significant improvement over the standard band ratio algorithms, there is a further need for value-added products that not only alert on bloom presence, but also differentiate on bloom type. Approximately 76% of the variance in the red and near-infrared spectral region is significantly positively correlated to chlorophyll A concentration. Strong assemblage-related spectral variability retrieved from high biomass bloom pixels can be exploited by the multispectral detection bands of ALCHI and MERIS. Information about the maximum line height retrieved at the 681 nanometer and 709 nanometer band was related to dominant phytoplankton types derived from phytoplankton count data, in order to derive thresholds which represent blooms that pose a high hypoxia or toxicity risk. The spectral signatures can be used to determine probabilistic ecosystem contextualized identifiers for waters dominated by either dinoflagellates or pseudonychia. These two images represent information derived from full-resolution Sentinel-3A ALCHI satellite data for 25 February 2019. The left image shows chlorophyll A concentration, whilst the right image shows the probabilistic phytoplankton community classification. Differentiated classes include dinoflagellates, red for very high biomass and green for lower biomass, pseudonychia dominans in yellow, mixed diatom assemblages of moderate concern in blue, and mixed diatom assemblages of low concern in pink. The locations of primary water intake pipes for abalone farms are indicated by blue diamonds. There is a good spatial coherence between very high biomass values observed on the chlorophyll A concentration map and the classification of dinoflagellates on the probabilistic classification map. Cell concentrations of over 2 million cells per liter, dominated by Goniolax spinifera, were measured at some of the aquaculture farm intake pipes. Although not without its limitations, this first-order determination of ecosystem contextualized thresholds assists in providing a simple, intuitive map-based indication of probabilistic phytoplankton-related risk to ecosystem managers and other stakeholders within the southern Benguela large marine ecosystem. I would like to thank RGNO team colleagues for the opportunity to present our research and development activities, and in so doing, opening the door for further collaboration between NatMerc and the CSIR.
Yes, no,
If there are any questions to Chris, please go ahead and ask your question.
did I put everybody to sleep? Too much like a lecture.
Krista, are you still recording the sulfur images, the imaging?
No, not really. And NASA's doing that anyway. They're producing a rapid fire archive, and it's still specifically for your region that got selected. And they've got a time series since the early 2000s up to the present, I think. So they do maintain an archive online. So whenever you need one, you can just go grab it.
Thanks, Bronwyn. Any other questions to Chris? It was quite a long and but a professional presentation, so I think you covered everything that you wanted to say.
I have a question about the 2018 bloom you said, where you were able to detect it from the satellite data. Are there any other instances where you've had something similar, or is that the only instance where you could relate what you see from the satellite and predicting that it's going to be a dinoflagellate bloom?
So it's pretty much a yearly occurrence for us. We have a bloom currently sloshing back and forth in St. Helena Bay. I haven't shown any of the more recent images, but because of the frequency of the occurrence, this kind of tool became necessary. And it wasn't just necessary because of the being able to detect powerful A already. The high biomass associated with our blooms doesn't allow the algorithms, the standard algorithms, to work at all. So we needed to develop something that can actually detect what we are observing over here. I think what Dion was quite interested in was what we was talking about towards the end, is being able to go beyond detecting high biomass, but also to differentiate between phytoplankton communities. And that's perhaps where I see our collaboration going. Um, I hope that answers your question. Sorry, I detoured a bit at the end, but I feel like
Thanks. Are there any other questions for Kristen?
Okay, I want to just thank everybody for coming together and listening to these presentations.